There are different theories of depression, and that is already clear. If there are so many different theories, that means that actually the pathophysiology of depression is not known. And I think this is something you already know, as I already described in the previous class. But we have to review these theories because that will all make a contribution to our understanding of depression pathophysiology and why it was so hard to develop very effective medications. There are, of course, treatment options for depression, but what is very good, what is very efficient, that would work immediately, that would, work in every patient, it's a matter of question, and it is not something we have today, and that is we want to have. And how we can get that is we want to discuss. We can get that only we can really solve the very complex pathophysiology of depression. I think you already have an understanding why it is so complex. These theories will help you further understand the effect of different mechanisms, but also it will be important for you to see that how the scientific process works itself. So maybe in the context of depression, maybe in the context of something else, it doesn't matter. Science is all about falsification. So you have different theories, you have experiments, and then we are going to see that some experiments are going to be actually challenged by other experiments. Some conclusions are going to be withdrawn because of some other experimental conclusions, and that is also going to be a real example in the case of depression pathophysiology from a scientific perspective. So the first theory is actually the Monoamine theory of depression. Yes, why do we know about this theory? This is again a little like a reversed way of making a theory, because how would you normally make a theory for something? You do some experiments, right? And then you collect some data, and then you understand the mechanism of something, and you make a theory. But in this case, it was not like that. It is like, you have the patients in the clinics, and then the clinics are, in the clinics, the patients are treated by different medications, and then you don't know anything about the pathophysiology of depression, but by some reason that drug works, whatever it is. And then people start to look the action mechanism of this drug, and as a result of that, they say, okay, probably that way should be included in the pathophysiology of depression. So the Monoamine theory of depression is that kind of theory that was made based on this kind of approach. So because of that, we say that since most antidepressant drugs act on the monoamine neurotransmission, and because of the, this very common observation in the clinics based on these medications that are modulating the monoamine systems, there was this, perhaps the oldest theory of depression came out to be the monoamine theory of depression. But now let's look at what are the monoamine system. What do we mean by when we talk about the monoamine? What do we mean by when we say monoamine neurotransmitters? So the monoamine transmitters are actually transmitters or neuromodulators that contain one amine group connected to an aromatic carbon ring. For example, here, this is a group called catechol group. You don't need to know so much detail about it, but I am sure you remember from high school chemistry that this is a benzene ring, okay? Benzene ring, which has two actually oxygen attached to it, okay? This is the catechol group, and then each of these major catechol groups are actually attached with an amine group. For example, here, ethylamine. In this case, here ethylamine plus there is this hydroxide. You see actually they are all derivative from each other. And then you have epinephrine. So the common point is that, for example, the catechol amines are called this because they have this catechol group in their structure. So one group of monoamine neurotransmitters are these neurotransmitters, dopamine, norepinephrine, and epinephrine, that are catechol amines. The other group is also serotonin, and the serotonin structurally is different than catecholamines. That's why they are not in the catechol group, but they are all monoamines because you see this is the amine group here. The amine group that is found here and here, here, here. There is a one amine group. That's why they are called monoamine neurotransmitters. Okay, this is the monoamine neurotransmitters, namely dopamine, norepinephrine, serotonin, okay, and also adrenaline or epinephrine. So this is also important. Please note that I don't expect you to know all these structures, okay? But what I expect you to know, in general, what are the common points between, you know, monoamines, why do we call them monoamines? And also I want you to know maybe that they are precursors. What's the precursor? So the precursor is a preliminary molecule from which some target molecules are being synthesized. For example, the precursor of monoamines is tyrosine. So you need tyrosine for these to be synthesized in the cell. The precursor of the serotonin is tryptophan. You need tryptophan. Tyrosine and tryptophan, what are they? Do you know? These are amino acids. Amino acids are building blocks of proteins. And actually, here's something very important. To realize that there are, you know, we have all an amino acid pool, but some of the amino acids that we have in our body cannot be synthesized by our body. These are called essential amino acids. And the essential amino acids are here: histidine, isoleucine, leucine, lysine, methionine, phenylalanine, threonine, tryptophan, and valine. So when you look at these you know, monoamine neurotransmitters, and when you look at their precursors, which of the precursor is an essential amino acid? Exactly. So that means that our body is not able to make its own tryptophan. That means that we have to get, with a good diet, we have to get it from outside. So otherwise, it's not produced very efficiently. Okay? So that is one important point. But where are those monoamines found? Where are they found? Where are they located in the brain? Any opinion? So we understand that there are some monoamine neurotransmitters. We understand that most of the antidepressant drugs somehow modulate noramine neurotransmitters. That's why there is a relief of symptoms of depression. And where are they found in the brain? Okay, again, this is a complex question for one reason. Brain is already very complex, so we want to know also brain is complex in terms of the levels of complexity. What are we talking where? What do you mean by where? Do you mean subcellular structures? Do you mean brain regions? Do you mean some circuitry? You see, you already know these things. You already saw these kind of different levels of complexity when we talk about the brain and its cells, its circuits, its molecules, its structures. Okay, so that's a little multi-dimensional question. So the first dimension may be the global level. Let's have a look in the brain level. Here you see the serotonin system. Here you see the norepinephrine system. In the previous classes, we have already seen that these are called diffuse modulatory systems. So these are modulatory neurotransmitters. There are also inhibitory and excitatory, the major fundamental neurotransmitters like glutamate and GABA. Then we learn these diffuse modulatory system. If you look at the serotonin system, here you see in the brain stem, in the midbrain, these nuclei from which all these serotonergic neurons originate, and then they innervate, for example, the neocortex, cingulate cortex, hypothalamus, thalamus, everywhere. Right? It is actually innervation to the brain is not restricted at all. It's very, very diffused. And here you see the norepinephrine system. Norepinephrine neurons are originating from this brain region called locus coeruleus, and then you see how actually, especially in the cingulate cortex, it makes innervations, okay? From there, it goes to the neurocortex, but you see the differences here. Here more, we see actually more serotonergic innervations compared to the norepinephrine system. And then to the cerebellum, and then it goes through the spinal cord to the peripheral nervous system, like in both of the cases. Okay, so this is two examples of monoamine neurotransmitters and their innervation to the different brain regions. Now, we just go a little down into our complex system and we want to observe where are they actually located. They are not located like these paths. They are located as molecules. These are the molecules, and the molecules are not really flowing like that. No, they are inside the neurons. But these signals that show the type of the neurons and neural connections, actually. There are some specific neural connections that have these neurotransmitters. So where are they found? Actually, they are found within the synapses. Synapses, as you know, the subcellular structures that are making connection between different kind of neurons. For example, this is a typical serotonergic synapse. What do we have in the serotonergic synapse? This is the first neuron, and it is axon terminal. And this is the second neuron, first neuron, and it's the axon terminal. And the second neuron, and it's dendritic tip. And in between them, this is the synaptic cleft. Okay, so this is the presynaptic cell. This is the synaptic space or synaptic cleft, and this is the postsynaptic cell. So here at this point, we are actually talking about two cells and their connection. That connection point, which is not a direct connection, you see there is a space here. That connection point is called synaptic cleft. So this is where the serotonin is found. This is how the serotonergic transmission is mediated in the synaptic level. So all these connections that I have shown you in this slide is actually in each bit is like that in the subcellular level. So here you see how many, again, you don't need to know all these details, but you need to understand certain fundamental things. That those neurons, each of them have this kind of structural subcellular structures, and there is, for example, here tryptophan. Remember, the tryptophan is the precursor of serotonin, and here in the presynaptic side, it has to be synthesized. 5-HT is the other term used to explain serotonin. This means serotonin. Okay? So from tryptophan, the synthesis of the serotonin has to be done. And that synthesis is is supposed to be catalyzed by associated enzymes. For example, if you look at here in the previous slide, as I already showed, it is also the same thing. From the tryptophan, 5-hydroxytryptophan is synthesized with the help of the tryptophan hydroxylase enzyme. So one enzyme, the second enzyme. These two enzymes are involved in the production of serotonin. And this happens here. So if you want to modulate serotonergic system, you need to modulate this, maybe. This is an enzyme. This is converting this from this. So this enzyme is mediating this conversion. So what if you block this enzyme? Then the whole thing will be blocked. What if you block this enzyme and the whole thing will be blocked? So it is all about the availability of serotonin. But the availability of serotonin is actually under the control of many different factors. You see here the whole thing. So the first is the synthesis of serotonin. And there are already multiple steps in that. So if you want to use a drug, so how it will actually, which of these molecular factors are going to be modulated by these drugs? Some drugs stop this, some drugs stop this. Each of them have a very different effect. Eventually, the final effect is actually to play with the availability of the serotonin between this and this. But how, to which extent, how long, all these things will have different impacts. It's a very sensitive system. As you already see, it's very complex. So the first thing in the serotonergic system is the synthesis of serotonin. The second thing in the serotonergic system is the metabolism of serotonin, meaning the breakdown of serotonin. So in the synthesis, there are these two enzymes. In the breakdown of serotonin, there is this monoamine oxidase. As you already know, monoamine oxidase A enzyme. Do you remember? We deal with the monoamine oxidase A enzyme genetic variations and its association with psychopathy in the presence of childhood abuse. So that is that enzyme. Because why? The monoamine oxidase A enzyme is a central enzyme that deals with the breakdown of all monoaminergic neurotransmitters. So that's like a famous guy here. Okay, so these are the molecular factors that is involved in the mediation or regulation of the serotonin system. So then anything that is making an impact on that, whether chemical, whether genetic, will have an impact on the serotonergic system. But it is not only those. What else? So the metabolism here takes place in these little organelles called mitochondria. Okay, here, but you don't need to know, just I explain what is it in this figure. All you need to know that there is a synthesis process, like something has to be synthesized in order to do its function. So it has to be synthesized, and once it is done with its function, then it has to be broken down. That is the metabolism. So between this and this, what do we have? Its function. What is its function? So you see here the serotonin is produced, okay, and then these are called synaptic vesicles. You have to think synaptic vesicles as cargo molecules, little cargo molecules that actually carry all these different specific, for in this case, serotonin molecules together like a package, and then they come to the presynaptic site, they fuse, they fuse with the membrane of the presynaptic site, and then they release their content into the synaptic cleft. Here, the high resolution of the view of what's going on here in this site. Here, when the serotonin is released, okay, then serotonin is going to bound the receptor in the postsynaptic cell. But not all serotonin will going to do that. Some serotonin is supposed to be taken back to the cell. So there are many different steps in the serotonergic action, and this is actually general step for all general mechanism, or like a common mechanism that you can see as available for all neurotransmitter systems. Just name of the molecules change. You may have, for example, glutamatergic system, just the name of the molecules will change, but the process are going to be the same. So it's not so complex as you think. Once you understand the general mechanism, then you will just put different neurotransmitters there, different receptors there, but the common mechanism is more or less the same. So there is synthesis, after the synthesis there is vesicle transport, and then the vesicular release to the synaptic cleft, and from there, the neurotransmitter is going to be bound with its receptor. That receptor is going to create some kind of effects, physiological response in the target cell. Okay? So, but now we are more interested in serotonin because serotonin is thought to be associated with depression. So what happens after that? The serotonin is taken back. So all these, any drug that is targeting in any of the component of this system is going to have an effect in the serotonin availability and serotonergic transmission. And that was the idea that we concluded, or people concluded, that serotonin should be involved in the depression. Because, why? For example, here, one famous antidepressant, fluoxetine, okay, is actually working in this system here. This is the one that takes back to the serotonin once it is released to the synaptic cleft. So one of the most prescribed antidepressant drug is working here. It is blocking the serotonin reuptake from the synapse. That makes serotonin more available to the target cell, longer. Another drug, for example, again, serotonin reuptake inhibitor. Another drug, for example, is actually interfering with this enzyme. Where is this tryptophan hydroxylase inhibitor? Where is that? Tryptophan hydroxylase. So, for example, if you have this, where is that? Yes. For example, some drugs, okay, this one and this one. So some drugs are just blocking conversion of tryptophan to this and blocking the production. There is a little mistake here. I will correct this later. But sorry, it is, yes, exactly. So think about the conversion of tryptophan to the serotonin. And there is this enzyme. What if there is a drug that blocks this enzyme that will block the production of serotonin? Okay, so that will interfere with the availability of the serotonin. Okay, some of these drugs here should be in this site, but for example, here, if you look at the reserpine, for example, if our theory that if more serotonin is actually inversely related with the depressive symptoms, that means if you decrease the availability of serotonin, then you should observe in the people that they are depressed. And actually, this drug, which is actually the blocker of the vesicular transport of serotonin from here to here, okay, this drug created a kind of depressant effect on the patients. So all these findings somehow led to the idea that serotonergic system, and the similar also with the norepinephrine system as well. I will not explain, but they also have similar but some different kind of molecules, but drugs that act in the synthesis, in the metabolism, in the vesicular transport, in the reuptake mechanisms of these monoaminergic neurotransmitter systems have an impact in the relief of the depressive symptoms. Okay, that's why there was this idea that monoamine theory of depression has emerged. This is one example of the that was used as an antidepressant. For example, imipramine. And then there was this idea that there is this somehow, as a result of all these clinical observations, there was this idea that there is somehow a kind of dysregulation in the monoaminergic system or diffuse modulatory system in the brain. And also that could also cause a kind of maybe, life stress, especially during development. As you know, during development, the brain neuroplasticity is very high. The brain can easily be adapted to any kind of effect, positive or negative. So maybe it could be because of that there is a kind of a monoaminergic dysregulation, and then it may cause the mood disorders. That's a question. Why? So far I described these kind of evidence that could be impacting on the pathophysiology of depression, but obviously that is not enough. So if all these things are okay, then why we don't just take it pathophysiology of depression explained by monoamine hypothesis, right? So then the story is done. We are okay. We should just go home, right? No. Monoamine hypothesis is not really strong enough to explain the pathophysiology of depression for several reasons. One reason is it usually takes weeks, quite a long time, for antidepressant drugs. So if there is a monoaminergic or monoaminergic dysregulation in the pathophysiology of depression, that means that, antidepressant drugs should immediately show their effect. But it takes quite a long time, actually, to get the relief with the patients when you use antidepressant drugs. From a clinical, that is for clinical, but from a biological or neurobiological perspective, you think that probably these drugs do not hit to the target or problem, the source of the problem, at the right source. It looks like it's an indirect way, somehow. Another thing, lowering the concentration of serotonin in synaptic cleft through the tryptophan depletion or serotonin transporter enhancer failed to induce depression in healthy subjects. So again, if it is related to serotonin system and serotonin dysregulation, then they can actually modify serotonin dysregulation in the healthy people, and they should expect a direct depression in those people, but it was the opposite. So in healthy people, when they have a dysregulation when they induced a dysregulation with the use of some kind of drugs, they did not actually observe depressive symptoms. And also, actually, some even recent scientific studies have led to the conclusion that maybe even high serotonin could be associated with depression. So far we always think that, oh, serotonin should be high enough, your serotonin is low, that's why you are depressed. But science doesn't say that. It is like very complicated story somehow. And here actually you see the evolution of theory related to Monoamine hypothesis of depression. Here the blue ones are showing the findings that support this theory, and the yellow ones show the findings that do not support this theory. As you see, starting from already 1970s, we don't have much evidence supporting this famous theory. Okay, so we just say that, okay, it may be related to serotonin after this class, but not directly. So, I especially would like to mention something about this, because that is very interesting. As you remember, maybe this guy from the monoamine oxidase gene variation, childhood abuse and psychopathy. So this is like, the person who did a lot of important studies for gene-environment interactions. And again, another textbook example for gene-environment interactions. But just for you to remember what is actually serotonin transporter, because the gene-environment interactions in this context is related to gene that is encoding for the serotonin transporter. Here you see a synapse. Okay, and in the synapse, this is a serotonergic synapse. You see, you remember there is synthesis, there is transport, and then there is vesicular release. And after that, there are postsynaptic receptors. These are all different kind of serotonin receptors. You don't need to know them. All you need to know that there is a presynapse. In the presynapse, there is synthesis, there is transport, there is vesicular release of the neurotransmitter. And then in the postsynapse, there is actually receptors that are targeted by the neurotransmitter, and that is a common mechanism for all kinds of neurotransmitters. Okay, I want you to know only this, not all these details, because this is an introductory course. But what you need to here understand, especially this, for our own discussion, and that is serotonin transporter protein here, this protein. So in the whole serotonergic neurotransmission, you can see here that the serotonin, when it is released to the synaptic cleft, is going to be taken back by the serotonin transporter, okay? So here, the topic in terms of gene-environment interactions and depression, and maybe for other psychopathologies, is related to this protein. Okay, now you know its function. So this protein is encoded by a gene. This is a gene that is encoding for serotonin transporter protein. Okay, this gene exists in humans in the chromosome 17. This gene exists in monkeys in the chromosome 16, and in the rodents in the chromosome 11. Okay, so we have actually homologs of all genes in the other organisms as well. So what do you see here now? As you remember, the most important thing that we learn in this class, that we all have alleles, genetic variations. So this is, for example, in this case of serotonin transporter gene, the gene encoding for serotonin transporter protein has two variations, naturally. Maybe this one variation is the short That means that the short version is, this is the structure of the gene. You don't need to know so much, but basically here there is this area. In some people, this area is shorter. In some people, this area is longer by nature. So if we take your, for example, blood sample, if we isolate DNA from your blood sample, and if we can easily understand which version you have for each of you. Okay, so some people naturally have this version, some people naturally have this version. I think this is a very interesting study. I mean, I really adore these kind of studies that actually made a kind of a real transformation in our understanding of behavior during health and disease. So that's again a very interesting study like the previous one in the first class. That's why I want you to pay 100% attention on this. So as I was mentioning, that there are two versions of serotonin transporter protein existing in humans. One is this one, the short versions in simple, and the long version. So what is the outcome of having a short version or long version? If you have serotonin transporter protein encoding gene in this form, that will end up the production of serotonin transporter protein in less amount. So you will have less serotonin transporter protein in your synapses. If you have the long version, you will have more serotonin transporter protein in your synapses. So basically this variation is associated with the amount of serotonin transporter protein. Okay, so there is a variation, genetic variation for the gene encoding for serotonin transporter protein, and that difference is in the level of protein level. So the people who has the gene in the short version is actually having the less serotonin transporter protein, and the others are going to have more serotonin transporter protein. What does it mean? That means that there will be more serotonin transporter protein if you have a long version of the gene, or less serotonin transporter protein if you have shorter version of the gene. Okay, and then of course people were curious what would be the outcome of this kind of having this kind of variation in the behavior. Of course, there are different kinds of experiments. For example, you can do a longitudinal study. What is a longitudinal study? In which you can just, in the Caspi's first study, in which they actually observed the boys from the age of 3 who were abused during the childhood until their actually adulthood. And then they genotyped also their monoamine oxidase A gene, and they found an association between monoamine oxidase A genetic variation and the abuse potential, severity of the abuse, and the committing a crime or psychopathy. These are one kind of studies. Other kinds of studies of the same question can be done in animals. Of course, in animal studies, you have much more opportunity. And in one study, what they have done is the following. So, here, this is the condition A and condition B. In the condition A, the baby monkey is grown with the mother, okay? Because maternal relationships since the birth is also important for animals too, like humans. And here, there is a maternal separation, and these do not have any maternal care. Okay, so these are two different conditions. You can take these conditions as environmental effect. Okay, what is the environmental effect on these animals when they are grown with this, like this condition A or condition B? And then they wanted to understand what is the effect of this condition in relation to genotype for the serotonin transporter protein. Okay, because the question is, as we already saw in the monoamine oxidase gene variation, for example, for psychopathy, we just know that some people, no matter what kind of bad, severe childhood neglect they have, they don't turn into a psychopath. So that means that environment actually does not create everything. If you have a, I don't know, very bad childhood, that doesn't mean that you are going to turn into a psychopath. There are many things behind of that, and one reason is probably the genetic factors that moderate the environmental effect. And that is again here the same question, how genetic factors moderate the environmental exposure. So there are two environmental situations. This is the mother reared, this is without mother. And then the genotype is, for example, this is written short and long because, you know, the short version or long version, this is a heterozygous genotype for the serotonin transporter protein. And this is homozygous. That means they both, because we have, you know, two versions, two copies for each genes that are inherited from both of our parents. So that could be homozygous, that could be heterozygous, okay, that could be dominant homozygous, that could be recessive heterozygous or homozygous. Here you have short and long as together as an heterozygous, and then homozygous long, long, short, long, long, long. Okay? And then here, what they do is the following. This is adenocorticoid hormone. This is a hormone that is related to stress response, okay? So that's a measure how you can actually understand the physiological response to the stress. For example, Arseni and Ibrahim, they look in the front and they look very cool always. They don't show much emotional reactions. So if something stressful happens, I would imagine that they would still be calm and cool. But some people, when there is, you know, even very simple thing, they are easily showing reactions. So that kind of thing is not really directly mediated with this hormone, but I just want to explain you that what is a stress response, how we react to things, how we can actually show reactions. And in the molecular level, in the biological level, one way to assess the stress response is to measure this hormone, okay? Because in biology, we try to find a way to quantify everything. There are qualitative things, good, bad, cool. For example, Ibrahim is very cool, looking cool, like Arseni is smiling now. And also ladies are smiling, Anna is smiling. But maybe they are very stressed, how would I know? That's a cultural thing. But in biology, if you measure these directly, that's a real objective measurement. That's why biological sciences and sciences in general are very much caring about the objectivity and measurement and parameters to quantify everything with numbers, with graphs, with plots. Otherwise, it's not objective. So we measure these hormones under stressful conditions, under the stressful condition. For example, this is a hormone of the hormone level before stress. This is hormone level after stress. Hormone level before stress, hormone level after stress. Hormone level before stress and after stress, okay? So if you look at this graph, what do you see? The first thing you need to understand that the individuals who have, let's compare this and this. The ones that are grown or reared without maternal care. And what happened here, if the individual has the short and long version, okay, of serotonin transporter protein, and if the individual or animal has the long version of the serotonin transporter protein. First of all, let's talk about what does it mean in terms of serotonin. If there is long genotype for the protein, that means there is more serotonin transporter protein for this case. And the animals who have this, they will have less serotonin. Is that correct? So the one who has more serotonin transporter are expected to have less serotonin, okay? And if they have less serotonin here, and look at their stress response compared to this one. The one who has more serotonin, which is associated with this genotype, actually are showing higher stress response. Okay? So that is, I think, very clear if you compare this and this. So, and then if you compare, for example, the individuals who have, with the animals who are actually having a maternal care during the growth period, the ones who have less serotonin, okay, are actually, they are pretty cool, huh? Before stress or after stress. But the ones who have the long version of serotonin transporter, which will lead to less serotonin in the system, they have actually more hormone reaction here. But that actually change, that is not a very, very significant change here between this condition and this condition. But between this and this is very significant. And also this and this is very significant. So look at this. So what do we understand with this experiment? We understand that actually we have different versions of the genes, and under normal conditions, it doesn't make us sick. Genes do not make us ill, whether you have this or that. The effect of the genes could become critical when you have an adverse childhood trauma during development. And then that could lead to this situation in which you have a dramatic response to the stress when you have this condition together with this condition. So that was idea, this experiment is interesting in terms of the serotonin availability, because normally if the depression is arising from the serotonin deficiency, why the stress response in these animals who were actually grown up in without maternal care, having, higher stress response. That is one of the studies that is challenging the monoamine theories of depression, for example. Besides, this is a study that shows the gene-environment interactions. And gene-environment interactions already determine how we will respond to stress and life trauma, events, everything. But gene-environment interactions already shaping us during the childhood. But how it is going to shape us during the childhood, again, depends on the genes and environment itself. It's not straightforward. If you have one version of the gene, maybe that environmental exposure, in this case, may not have a very strong impact, like this. Look at this and this. And under the same environmental condition, which is not a very, which is an adverse environmental condition in this experimental model. But what happens is the stress response on this, if you have this genotype, the stress response is not as much as this case. Although the same environmental adversity was the case. Here, especially during the development, is essential to understand. You can have environmental adversity when you are an adult, okay? But that is more significant when you have an environmental adversity during development. Why? The brain is highly plastic and easily adaptable to effects from the environment. But that doesn't mean that you will not be impacted by trauma when you are an adult. Of course, you can be impacted. So this study is an interesting study for several reasons. The first reason is that because it's a study that is challenging the monoamine theory of depression. The second important factor for the study is that this study is explaining the gene-environment interactions, that it is not like, oh, you have less serotonin, so that's why you are depressed. It's not so straightforward. Actually, the most fundamental thing for you, I want you to understand in this class, that there are genetic variations, there are environmental adversity or conditions during the development. The genetic variation is huge. Here we are only talking about one gene, but normally I talk about more than 20,000 of genes that have little effects, and all their variations and all the environmental exposure. That's why we have different reactions to the different conditions. That's why we are all unique. Okay, so that is just here one example for one gene. To study all genetic variations all together with environmental factors should be more systematic way of understanding these kind of patterns, but actually we are on that way also. We are on that trend. We are trying to, there are ways to study the whole genetic complexity of all different genes at once and compare it with the environmental factors, which is called exposome. So you have genome, you have epigenome, you have transcriptome, okay, you have phenome, which are all these things. When I say genome, phenome, that means we are not talking about one gene, that means we are not talking about one phenotype, but we are talking about multiple things at the same time and compare them systematically. So don't forget that here just one example with one gene and with one condition, just to understand you the relationship. But actually it is thousands of even more genes and their variations and environmental interaction. This is actually an example for you to see how we look at the study of pathophysiologies, the gene-environment interactions, but I will not go too much into this detail. I want to show you another reason why monoamine hypothesis. You know, monoamine hypothesis is important for us. Why? Because to see that how a hypothesis can be defeated. So it's a good example for scientific critical thinking for you to practice. And the other is that how actually the theories of depressions are evolving from one stage to monoamine neurotransmitters to probably maybe glutamatergic system. Because now we have a completely different picture, and the future drugs of depression may act on glutamatergic system instead of serotonergic system. Maybe we will all leave eventually once, because there is a huge effort in the scientific circles to find the new drugs, new, strong, good drugs. There is and there is some hope to find those new drugs, and those drugs are definitely not targeting monoaminergic system. They are targeting something else, and what is that? That is very interesting. Here you see this is a glutamate receptor. These are the glutamate receptors, and obviously this is a glutamatergic synapse. This is not a serotonergic synapse. This is not a dopaminergic synapse. This is not an norefinergic synapse. Okay, this is a glutamatergic synapse. So what do we have here? Here you have vesicles that are filled with the glutamate neurotransmitter, okay? Normally what happens in the synapse, if this neuron is firing, it will release glutamate to the synaptic cleft, and those glutamate molecules are going to bind to its receptors. As you remember, there are two kinds of glutamate receptors, Nmda receptors and Ampa receptors. As you remember, Nmda receptors were very important for learning and memory. So here, then there is another neuron here as an example. So you have to think that a neuron is actually here, you see 1, 2, 3, just a bit of 3, but normally it's much more complex. But here there is a little story for you to realize, and that story is, this is one neuron, an inhibitory neuron, let's say, and this is actually another neuron, which is glutamatergic neuron. And this is, inhibitory neuron means it's a GABAergic neuron. And then here you have this normal situation. And then there is this drug called ketamine. And what happened here, when ketamine is actually acting here, so the ketamine here is actually somehow blocking this neuron to impact on this neuron. What does it mean? This is an inhibitory neuron. If this inhibitory neuron is active, it will not inhibit this neuron. If this neuron is not inhibited by this neuron is going to release its glutamate. So basically here, this is actually somehow stopping. This is a neuron that is pushing this neuron to stop glutamate release, okay? But if you stop this neuron doing that, which is done by ketamine, ketamine was actually here, the ketamine receptor, which is a kind of a receptor, Nmda receptor is modulated by ketamine. And then once this receptor is blocked by ketamine, then this neuron becomes inactive. And then this will, if it becomes inactive, this neuron will be more active. Okay? This is what ketamine does in the synaptic level. Actually, this is also somehow a proposition. It is not maybe there are some variations in this proposal. But why did I mention that? For two reasons. The first reason is that when you give ketamine to people who are depressed, and especially those who have a treatment-resistant depression. Do you know treatment-resistant? No matter what, it doesn't work. They don't get better. It's very hard. In a few hours after the take of ketamine, those depressive symptoms are gone. That was a huge discovery that shook the scientific circles for two reasons. Okay, if ketamine makes such a strong impact very quickly, that means that, first of all, ketamine is acting on a system that's completely unrelated to monoaminergic system. That means that's a very important finding for the pathophysiology of depression, that probably it is related to some other systems than monoamines. The second, that's a hope to find new drugs that act very rapidly. Okay, but we cannot just use ketamine as an antidepressant drug because of its side effects, because of its abuse potential. Normally, it is used as an anesthetic, but it has this kind of effect on the patients. So that's why there is now, and its effect in the cellular level is this, as I already explained. And that is another reason now we are thinking that probably depression pathophysiology is not really related to monoaminergic depletion or dysregulation, but it is related to maybe this kind of modulation. Why? Because its effect is much quicker, in a few hours. So people who are depressed, they use antidepressants. It takes at least 3 weeks to see if it works or not. So I tell you the trend. You go to the doctor, they prescribe you a drug, and they tell you, you need to wait 2-3 weeks to see if you get progress. And that's a huge time. First of all, nobody has 3 weeks time. I mean, you wait 3 weeks to see, and then you go to doctor and then say, does it help? I don't know. It's like, so is it going to be like a kind of adjustment with the dose? Is it going to be like a new adjustment with a new prescription? How it will be? It's totally like based on the talent of the doctor. There is no enough objective ways to actually evaluate the situation, and that's not a very optimistic situation. And think about now that you give the drug and in 2 hours the patient is totally fine. So that's amazing. But of course the ketamine itself is not useful as a pharmaceutical agent because of its abuse potential, because of its side effects. But now, because of this situation, there is a huge effort to find out ketamine-related or ketamine-like substances which do not have the same side effects like ketamine, but induce the same antidepressant effect with ketamine. Okay, and that is actually the age of or wish of rapidly acting antidepressant drugs. So we are now in the situation in which there is an effort to find out these kind of drugs, and there is a paradigm shift now in the pathophysiology or theories of depression from this monoaminergic theory to this kind of focus. Now, the monoamine hypothesis, as we already discussed, is a little old-fashioned. So it's not like, so you don't want to talk about like in the magazines, oh, your serotonin is low, that's why you are depressed. No, We don't say that. I think we have enough reasons not to say like that. Okay? So that's why there is a huge effort in this subject to find out rapidly acting antidepressant drugs. So for those who suffer from depression, I want them to be hopeful, because I think there will be a huge progress in the near future in the both understanding and also both in the treatment. We know that there was a huge bottleneck against the development of new drugs for psychopathologies. We know why, because of the huge complexity. But one by one, these complexities are being resolved. One of them that we discussed already previous class, what was it? It was actually the identification of genes that are associated with depression as a result of genome-wide association studies. Until recently, we didn't know which genes were related. There were all these candidate genes, and they were never actually reliably validated. So it was like all question mark, like the serotonin hypothesis. It was all about question marks. But now, little, one by one, there are real, validated, reproducible results that could take us to a better point from where we have been before. So, until now, I was explaining you Monoamine hypothesis, how it was so fashionable before, and then I have shown you that some findings there are some findings that give the opposite results in terms of the serotonin dysregulation and depression. And they basically challenged the monoamine hypothesis. Another idea that challenged the monoamine hypothesis is the findings regarding the action of ketamine in the central nervous system and the relief of the depressive symptoms. So all these findings made us think, okay, we leave the monoamine hypothesis alone. We change our focus, and that focus is probably the glutamatergic system. But the issue is not so complex, so you really need to know all these different theories of depression. And the other theory is diathesis stress hypothesis. Diathesis is vulnerability. Vulnerability stress hypothesis, and this is a general idea I have always mentioned in my classes, and that is, you have a genetic variation or combination of genetic variations that makes you vulnerable to adverse environmental effect. That is the stress during development. And then in the adulthood, you have a certain kind of psychopathology depending on the vulnerability you have. Each of us have our own vulnerability maybe. So for that reason, of course, for the depression, there is also this kind of hypothesis that we have already seen that in the gene-environment interactions with the animals, with or without maternal care, you remember? So there is this case of the, genetic variation and environmental adversity, how it could make an individual vulnerable. With the animal studies that I shown in the slide, this one is actually can be also seen under the title of vulnerability stress hypothesis, that there are genetic variations, and that genetic variations could actually moderate our stress response, and some individuals could be more vulnerable to stress compared to other individuals as a result of all these interactions. So actually the vulnerability stress hypothesis is such a hypothesis, but it mostly converges on our stress system or stress response. Stress response. What is stress response? When we think of stress response, you have to think maybe some major components. The hypothalamus, okay, and then the pituitary gland and the adrenal glands that are found in the kidneys, in the top, okay? So these are the main structures that actually mediate the basic stress response. Stress response is a natural phenomenon. It is good because it helps us survive. But when it gets out of control, it can become a pathology. Okay, so it's all about, actually it's a natural response, but it can be chronic, then it can be devastating for the individual's health. all right? But here especially, remember our amygdala. Here you see the coronal section. This is the amygdala here. Here the basolateral nuclei, this is the central nuclei of amygdala, central nucleus. The point here that this basolateral nuclei receive, so you see me, okay? And that is a sensation, vision. All the sensations, everything from sensory neurons reach to the brain and they can come to the thalamus, and the thalamus send it here. So you see something, a mice, little mice that is, or snake, maybe there is a snake here. Then suddenly we would show a reaction like, we will be aroused, we will be scared. We are not scared of cars, do we? Are we scared of cars when we see in the street? Why are we scared of snakes, but not cars? If you make a statistics, people die more because of car accidents. Nobody died today because of snakes. But we have evolved in such a way that snakes, we generate a kind of fear when we see them, but we are never scared of cars. But in reality, cars are associated with more death of humans. So it's all about how our brain is evolved and how we sense certain things dangerous, and how all this information is traveling to the amygdala from the thalamus and cortex, and especially the neocortex. And then amygdala is sending all this information through the basolateral nuclei to the central nucleus. And from there, look at the projections of amygdala. Hypothalamus. So here, here you see the central nucleus of amygdala is sending the signals everywhere. I'm showing actually the fact that how amygdala is important for the stress response, because it's a central nucleus that is sending hypothalamus the signals. Hypothalamus, as I showed in the previous slide, is the one that is involved in the stress response. But hypothalamus is itself under the effect of amygdala and its innervation, okay? And the hypothalamus is the one that activates the stress response. But that is in the brain, so if you see something, if you're scared of something, you're gonna have a kind of a stress response, meaning that maybe, you know, your heart rate is going to increase, you start to sweat, depending on the individual as well, but also in the organism itself, there are different kind of pathways that will be activated. The organism is set to an alarm condition. Then the other factors that are related to your survival will be maybe a little suppressed. For example, digestion is going to be a little suppressed because there are more urgent conditions. There are more emergency there. And so the metabolic activities and everything is going to be adjusted optimally for you to assess the threat and respond. And your brain and everything is going to adapt to that. That's a stress response. So it's not like you are scared, but there is a lot of things going on in your body at that time. And here, the amygdala is actually very important in that. You know, they have done some studies in orphans who are grown up without maternal care. They found that their amygdalas are bigger than the normal kids in the adulthood. So think about that, big amygdalas, like, you know, responding. But again, it's not so straightforward. All these things are the result of cumulative studies, and with every individual, it again depends on genetic variation and environment as well, even within the orphans itself. So, but what do we have in the stress response? What do we have in the stress response? So basically, amygdala is the one that is sending the signals to the hypothalamus here. And once the hypothalamus is actually stimulated, it will send corticotropin releasing hormone, and then it will stimulate anterior pituitary gland to release this ACTH hormone, and then it will actually goes to the blood, through the bloodstream to actually stimulate the adrenal gland that are found in the kidney to release cortisol. Cortisol is a stress hormone, and once the cortisol is actually a kind of physiological signal that actually makes the body to be primed for this extraordinary situation, the stressful situation. So the heart rate is going to increase, other kind of survival-related factors of the body are going to be maybe more suppressed for a while. There will be more glucose arrival to the brain. So the other things will be a little put on the backside to focus and use the body for the optimal, rescue, for you to rescue from the threat, perceived threat. So that's a good thing on one hand, but on the other hand, so this cortisol is a group of is a kind of glucocorticoid here. So there is, this cortisol is going to go back to the brain, and there is a kind of negative feedback. What do I mean by the negative feedback? So it's like a one cycle, and once the, everything is done, and then eventually this hippocampus, for example, here is going to get this response from these molecules, and it will get actually give signal to hypothalamus, stop, enough, no more stress. But if there is a problem in the circuitry, if this, for example, if there are no enough receptors in the hippocampus for some reasons to receive this feedback, negative feedback, this is a negative feedback. So from here, it goes back and it should stimulate this to make this stop. But if this is not sensitive enough to this negative feedback, which could be the case for some people for different reasons, what are those reasons? Genetics, environment again. Then this feedback loop doesn't work, and then there is an overactivity of the stress response, which is not a normal response. That could happen because of many reasons. Sometimes even in the adulthood, if you are exposed to chronic stress, eventually your system could break up. And then you may end up with such a situation that this cycle doesn't work properly anymore, and then you are overreactive and you are living in a very, stressful situation, which is also stressful biologically for the body. Here is actually how this is the HPA axis. This HPA axis is this system: hypothalamus, pituitary, and adrenal axis. Hypothalamus, pituitary, adrenal axis is called HPA axis. Okay, and the HPA axis receives the signals in a positive way. That means it stimulates this system. From amygdala, it receives such a signal, and from the hippocampus, it receives a kind of a negative. That is the source of the negative feedback loop. But if there is a problem here, that will not happen. So that can happen, that problem can happen due to several reasons. Of course, mostly, you would say, gene-environment interactions during especially childhood. Then you have already a situation like, for example, those monkeys which were grown without maternal care. I could imagine that maybe their HPA system also is adapted in such a way that their negative feedback loop somehow does not work very well. And then that is a vulnerability model, that the brain is vulnerable. The brain is vulnerable to stress, and the brain is ready to get a pathology once there is a serious stress in the adulthood. Okay? So this could happen for depression. This could also happen for anxiety disorders as well. Especially for the anxiety disorders, we have this kind of stress response problems which cause people develop a kind of problem which is associated with the management of anxiety and stress response. So when this stress response is out of control, people get anxious. So this actually vulnerability stress hypothesis can be perceived as a general hypothesis for different mental disorders, including depression and also anxiety. So what makes stress response different from one individual to another? The only thing you have to say, genetics, life experiences. Very simple answer. Genetics, why genetics? Because there is genetic variation, and that variation is unique in every individual. And that variation is not just with one gene, it's like, numbers of genes that are expressed in the brain, and those variations. It's huge. There is a huge variation. Look at us, how we are different from each other. The brain and the way how brain works is also different because all these genes different in the molecular level, all these synapses, how they respond to serotonin because they have longer version or shorter version in the transporter. And think about this in the zillions of neurons with zillions of molecules. Think about all these physiological variations. That's huge. And then life experiences are so unique for each of us. Culture, economy, personal situations, and everything. That's all unique. Plus, it's not really straightforward. Oh, you have a very adverse childhood. That doesn't mean that you're going to have definitely a psychopathology. That is moderated again by genetic variation. Maybe your genes somehow make you less vulnerable. That's why you still survive. All right, so I think after all, I always systematically repeat genetic variations, life experiences, and their interactions for everything in this class. And I am sure you get enough of it. And that's enough for today. See you next class.